Just by way of a little bit of background about the IMI2 scheme, this is an EU European Commission overseen project that brings together industry and academia and other stakeholders in a pre-competitive collaborative forum. The idea is that it must focus on unmet needs, and it is based around competitive calls for proposals going out through peer review and, as I'll show you in a moment, a fairly robust series of assessments. The total budget for this from the public sector side is approximately 1.5 billion euros, and this is matched via the EFPII partners with industry in-kind and cash contributions. So the overall program here is of substantial funding, and the intention is that it should be addressing major issues at a transformative level. The assessment process is time-consuming and robust, but as we've successfully gone through it, I think very fair. The initial step here is this topic definition, and here industry and the European Commission come together and identify key challenges. A public call goes out, and consortia from both academia and industry are invited to bid to address the specific challenge. After the first stage, one single consortium goes forward into the second stage of this process, at which point the initial industry partners who commence the process work with the applicant consortium to finalize the application, which then goes through a further round of peer review and hearings in Brussels before the decision for a grant award is made. Just as Roberto has outlined, and we've heard very eloquently this morning, there's a clear imperative for biomarkers. We have this paradox with a large proportion of the population affected with fatty liver disease, but only a minority going forward to a significant morbidity or mortality. And as we've already heard, there are a number of challenges here. We need to address biomarkers that are diagnostic in terms of grade and stage of disease, prognostic in terms of identifying those individuals who may be rapid or slow progressors, and also monitoring biomarkers that can actually be used to track individuals during the course of disease and to follow treatment response. And so these are the same challenges that Litmus intends to address. It is, of course, true that this lack of tractable biomarkers has already impeded the diagnosis of patients in the clinic and their care, and it has also caused a great challenge in the conduct of clinical trials. And so this is the background that we must address. What is the Litmus concept? Well, first of all, it's that a goal-orientated, tripartite collaboration bringing together the end users of biomarker technologies, be that clinicians or pharmaceutical industry, independent academics, and biomarker researchers and developers. By bringing these three groups together, we'll be most effectively placed to address the challenges that we face. The intention is that Litmus will implement a robust technology unbiased platform to conduct the systematic study and validation of a broad range of biomarkers, both imaging biomarkers and wet laboratory-based biomarkers, as well as near-patient testing technologies. And all of this will be conducted with reference to fully adjudicated liver biopsy data, for which we already have a team of pathologists who've been reviewing our biopsies for a number of years, as I will explain. The intention is that the data collected and the processing of all samples will be aligned with the EMA and FDA expectations so that we can optimize the quality of our outputs. And a very early part of the Litmus process will be to engage with the regulators to begin the constructive discussions with them about what it is they would expect us to deliver. Finally, our ultimate goal is to establish a defined set of biomarkers that either singly or in combination will enable the detection and monitoring of disease progression or regression from fatty liver steatosis through to steatohepatitis, fibrosis, and cirrhosis, both for use in clinical practice and drug development. These are lofty goals, and one of our unique advantages is that we already have strong collaborative foundations in basic science and discovery science. The Litmus Consortium is the direct descendant of two previous rounds of European Union funding. First of all, the FLIP Consortium, which was €6 million and ran to 2013. More recently, the EPOS Consortium, which Pierre has already mentioned briefly, which is currently driving forward on discovery science and multi-omics approaches. And now Litmus, which is funded for €34 million, will run for the next 5 years, addressing these major diagnostic challenges. We also have an established registry of patients, which has been recruiting now back to 2010, both in member sites who were parts of FLIP and EPOS, and a number of additional centres also bringing in all their data. And for Litmus, we will expand this further using a hub-and-spoke model across, I think it's 13 countries across Europe. I'm also pleased to be able to mention our strong collaborative links with the NIMBLE Consortium, particularly Drs. Sanyal and Harrison, which will allow us to foster even stronger transatlantic collaboration. All our samples will be centralized to a single biobank in Luxembourg, and all data will be collected using a single web-based platform, which has now been running very successfully at centres across Europe for a number of years. The NAFLD registry already brings together significant clinical data sets regarding anthropometrics, medical history, and so on, adjudicated histology with digitalized images, and an extensive biobank resource. But off the back of FLIP and EPOS, many of the cases that we still have under follow-up have also had an integrated omics dataset generated, including SNP variation, liver transcriptomics, DNA methylation profiles, lipidomics, and metabolomics, and this data will also be available to inform the work. And of course, because our registry runs back to 2010, many of the cases have been under follow-up for a significant period of time, allowing us to look towards those all-important hard endpoints that we seek to address. That's just a very quick screenshot of the web-based registry, which is pulling patients together. And just to give an example of the range of data that we'll be collecting and also the range of samples that will be collected, this is a snapshot of the selection of samples that will be collected at enrollment. One of the key challenges with a registry, and particularly when you're pulling together data from a lot of centres, is the pre-analytical quality of the samples that you will be using. And this is something that Litmus and EPOS have recognised for a long time. And so all our centres have been using standardised approaches in terms of sample collection and handling that we hope will minimise the variation in this setting, along with a rich data set capturing, for example, patient symptomatology, dietary behaviours, and lifestyle in general. And so all of this data is also available to us to feed into the study. No project like this could be achieved without a substantial amount of collaborative work. We're very fortunate that, as you saw on my very first slide, there's strong links between the academic partners and the industry partners. Dr. Julia Brosnan from Pfizer is the project lead, and I will serve as the project coordinator for this. And as you can see across each of our work packages, there's strong leadership from both sides of the fence, which we're slowly pushing down to the ground. So to conclude, Litmus is a focused, pragmatic and goal-orientated programme founded on a strong track record of NAFLD research and discovery science. Our ambition is to make a fundamental difference to the way fatty liver disease is diagnosed and clinical trials are conducted. And we believe we have a demonstrable capacity to address these challenges and provide much-needed clarity for the biomarker discovery at scale and pace. It just leaves me to acknowledge all the partners who are taking part in Litmus and to thank you for your attention.
Thank you.